Hello, and welcome to this Pharmaron podcast, part of our DMPK Insights podcast series. Thanks for joining us. My name is Chris Bode, and I work on all types of DMPK studies as well as BCS classification studies performed by Pharmaron US Labs based in Exton, Pennsylvania, near Philadelphia. Today, I'm happy to be speaking with Peter Turnbaugh. He's a microbiologist and a professor of microbiology and immunology at the University of California, San Francisco. Welcome, Peter.
Hi, good to see you.
Great to be with you. So Peter is in the middle of so much of the groundbreaking research in a field that I find incredibly fascinating, the gut microbiome. And in the interest of full disclosure, I am absolutely not an expert on the gut microbiome, but it's probably my favorite scientific topic. I follow it kind of as a hobby, always with the hope that I'll get to work on it at some point. And although I think it's been a really hot field for a decade or two, it seems like it's still like it kind of flies under the radar still. So I'm always trying to let people know about it. And I've organized 2 symposia on the gut microbiome under the banner of the Delaware Valley Drug Metabolism Discussion Group. In fact, that's how I met Peter. He helped me organize the first one in 2016. Before we really get started, I wanted to make sure I didn't forget to let you know that Peter hosts his own blog called Science is Fun with an exclamation point. You can find it on Apple Podcasts, maybe other places too. And Peter, I'm so glad you told me about your podcast for a couple of reasons. One is this is my first podcast, so I took some comfort in knowing that at least you've done this before. And the ones I've listened to are really interesting, and I've learned a lot. So I know on your podcast,
Mhm.
the episodes are very long.
Oh yeah, maybe too long, I've been told.
I don't know. They're just packed with information, and it's really interesting. It seems like you I don't know if this is true of all of them, but the ones I've listened to, it seems like you lean heavily on your UCSF colleagues.
Yeah, I mean that's not intentional. I try to reach out more globally. We've had some international guests. Fergus Shanahan was on. I tend to do better with microbiome researchers that are in other places. Then once I get in a different field, I really, they typically don't reply to me unless they're at UCSF.
Yeah, no, it's been great. I've learned a lot about things that I really never thought of before, like the placenta.
Yeah, yeah, really fun.
This is so fascinating. So, for our listeners, there's so many fascinating things about the gut microbiome, and I'd like to share a couple of intriguing facts. First, when we're born, we're 100% human, but within a few years and for the rest of our lives, we're more than 90% bacterial cells. There are 10 times as many bacterial cells in our bodies as human. And second, if the gut microbiome were an organ, in terms of the total mass of bacteria, it's as large as the liver. So those are just a couple of things that I found really fascinating and got me wanting to know more about it. So Peter, can you talk about how you got into this field, kind of your origin story, I guess?
Yeah, definitely. So I went to grad school at WashU in St. Louis. Really excited about genomics. So it was, at the time it was like, shortly after the Human Genome Project, and there had already been some isolate bacterial genome sequenced. And so I was just really excited by the potential of being able to, see all of the genes within an organism. And so, yeah, I didn't really have a biological question in mind. I just kind of went in wanting to do something related to genomics. And was also really fascinated by genetics and, how genes work. And, fortunately ran into Jeffrey Gordon, who's, one of the really early pioneers in this area. And he got me into it. And the timing couldn't have been better. It was around the same time Ruth Lay joined his lab, and she brought all these culture independent methods from Norm Pace's lab, and so we worked closely together on a lot of the early studies in the group.
Yeah, I wondered if you had been in Jeffrey Gordon's group. Obviously, he's one of the foundational people. And you mentioned Norm Pace. I was surprised when I read that his name keep coming up in the gut microbiome. I actually took he was one of the lecturers in the biochemistry course I took at the University of Colorado Medical Center. I was doing
Yeah,
my
he's had a
PhD.
huge impact.
Yeah, that's fascinating.
Yeah, I mean the big, as you probably know, the big revolution was this, the use of PCR, of specific genes like 16S, to give you a profile of a microbial community. Before that, people were using, really old methods that you've probably never heard of, DGGE and Refleps and, other things to, get kind of fingerprints. But you really didn't get any information into what type of microbe it was. It was just all kind of abstract.
Yeah, that's really that's really great. I mean, like I'm not a microbiologist. I'm also, um, the genome. I'm kind of on the outside of that too. But I
Mhm.
quickly you appreciate how important it is to the genomics is and metagenomics, I guess you often call it, right? Because it's not really it's in the host, but it's not the host that you're sequencing.
Yeah, and I think like nowadays people, at least in the field, don't really appreciate how amazing these culture independent methods are. Really around the time I started in grad school, a lot of people were still culturing, and it's just so hard. Even if you are able to culture the microbes, the culture condition you choose really skews the community. And so you get really misleading answers. Like you might think that E. coli is the number one bacteria in the gut. And so being able to just like take any sample of interest and use these sequencing methods was really a big step forward.
Now, are all the ones that are really important in terms of the gut microbiome are they all anaerobic?
Yeah, so that I mean that's one of the big challenges. It's been largely solved by the use of these. We work mostly in these chambers that allow you to control the gas. Um, and so but if you're trying to, definitely if you're trying to culture on your benchtop, you'll lose the vast majority of microbes that are in the gut.
Yeah, I want to get back to that later. Just in terms of practical considerations for studying bacterial drug metabolism. And
Mhm.
I know there's so much more about the gut microbiome that doesn't have anything to do with drug metabolism, but this is kind of
Right.
it's a
Yeah. Mhm.
DMPK podcast series. And I have to say, initially, um, I was skeptical whether gut bacteria could really be important in terms of drug metabolism because I thought they were only in the large intestine, and most drug absorption takes place in the small intestine for most drugs. But clearly, gut bacteria are important for metabolism of some drugs. And does that mean that there are a lot of gut bacteria in parts of the small intestine?
Yeah, that's a great question, and this is a question we get all the time. I think if you look at a textbook, there's kind of the classic um, chart that'll show very low levels of colonization, in the duodenum, the beginning of the small intestine, and then as you go throughout the small intestine, the density gets higher and higher until you reach a peak in the colon. Um, that I think it's important, or I guess what I've learned getting deeper and deeper into this area is that, those kind of textbook diagrams are a vast oversimplification of a much more complicated picture. Um, and so, in reality, there can be, some individuals that have much higher levels in the small intestine. You might have heard of something called SIBO, which is small intestinal bowel overgrowth. So you definitely see patients that can even reach much higher levels. And then you can also reduce levels in the colon due to certain insults like antibiotics or diet, other factors. And there's also, I think, the big theme that we've learned in general is that the microbiome is incredibly variable between people. And so even though, on average, that kind of picture holds up, um, it's not true for everyone. I think really what we knew from the old literature, but I think people have been, I think rightly really fixated on the the massive levels of colonization in the colon, which reach like 10 to the 11 cells per gram. So that, is obviously important given how abundant the cells are. But that doesn't mean that the stomach and small intestine is sterile. There's always been microbes in the stomach and small intestine, and that dates back even to the early studies. And has led to some really big breakthroughs, like the study of Helicobacter pylori.
Yeah, H. pylori in the stomach. So, would you say that the gut bacteria are important for drug metabolism, particularly for drugs that are not well absorbed? So, there's still the parent compound is still hanging around lower in the small intestine, or is
Yeah, that's a great point. This is mostly work that we haven't done with other labs in pharmacology. I think as we've gotten deeper and deeper into pharmacology, have learned that as you know better than I do, the kind of ADME properties of drugs are a lot more complicated than I typically imagined. And so, some drugs actually stay within the GI tract very long. We're doing a lot of experiments right now on rifaximin, which is a drug that is considered to be basically not absorbed at all. And so the whole drug gets excreted in the stool, absent a tiny fraction that gets absorbed. So that can definitely happen, and that gives you potentially much higher doses of drug than what's delivered in the colon as the lumen gets concentrated due to loss of water. And then in addition to that, a lot of drugs are released back into the gut after they're absorbed, and so that's something we think a lot about is kind of the classical drugs that are subject to enterohepatic circulation, but also, more novel kind of suggestions in the literature that drugs might, more drugs might kind of go through that route than we typically think about.
well, I know that's there are issues with cancer drugs, for example. Irinotecan is one that it's glucuronidated in the liver, it's excreted into the bile, it gets into the gut, and then for bacteria that will deglucuronidate it. Basically, that's fuel for the bacteria. But then you've got the parent compound that's really toxic. And I know Matt Redenbaugh had done some work on which I thought was fascinating, to address that, not with an antibiotic to kill the bacteria, but with a selective inhibitor of the bacterial enzyme, the beta glucuronidase. But I'm sure there are more examples like that.
Yeah, definitely. One related to that, we're very interested in fluoropyrimidines, which are a common drug used for cancer. They're not, those are not drugs that we typically think of as, going through, glucuronidation and the same release back into the gut. But they do have a lot of GI toxicities. We have a preprint out if you're interested, kind of showing that changing the microbiome can affect the intestinal levels of the drug due to an enzyme that we showed inactivates 5-fluorouracil. And we think that's important for kind of controlling the local toxicity of 5-FU. Interestingly, 5-FU and irinotecan are given together in a cocktail. So, you can potentially have like bacterial effects on multiple components of these cocktails that are given to cancer patients.
Oh, yeah. Okay. Very interesting. So probably like with irinotecan, probably some some patients have the bacteria that well, I don't know. Maybe everybody has the bacteria that would deglucuronidate that, if it appears, if it's excreted into the gut. But probably like different people do it to different extents, and maybe even the same patient at different points in their life would probably handle it differently. Is that fair to say?
Yeah, that's a great point, and definitely like kind of the direction we want to go, in kind of our long-term view of this area is, trying to take more of a pharmacogenomic or pharmacogenetic approach. I think that we're still at the point in the field of just kind of identifying what the main players are. It's really helpful in the case of you mentioned the Gus enzyme or beta glucuronidase, or in the case of 5-FU, pre-TA, or which is a bacterial version of dihydropyrimidine dehydrogenase. Just finding those enzymes we think is really important, because what we and others have found is that they don't typically kind of fall within a single group of bacteria. And so, if you just know the names of the microbes, it's really hard to predict whether or not they will metabolize the drug. Whereas if you know the enzyme, that is a better signature. And so, this is a bigger issue than, human genetics, where, we all basically have the same genes. We just have different versions of them. In bacteria, you can have two closely related microbes where one doesn't have the gene at all and the other one does. And so, it's very hard to kind of infer what bacteria are doing just from their name.
Yeah. Okay. And then I guess, I mean, in order to do that, to search for in different bacteria, is there a sequence that you can look for and know, okay, this is going to be part of the gene for this enzyme, or are they different enough across bacteria that you couldn't even look for that?
Yeah, like now, once you kind of find a gene of interest in a single organism, then what we love to do is, go mining through all the genomes that are out there. And that's really interesting. We often find, and I'll go back to Gus as an example that Matt's lab has really nicely worked out. That those genes are found in many different microbes. And just to add like another layer of complexity, what he's really nicely shown is that they actually, even though they're similar to each other, when you look at the gene sequence, the proteins have different folds. And that seems to matter for the types of compounds that they can deglucuronidate. And so, even though it's the same kind of host modification of chemicals, he thinks that these different Gus enzymes can accept different types of substrates. And so, one might be really good at, like the enzyme from E. coli is really good at handling the irinotecan metabolite, whereas others might be better suited at acting on glucuronidated compounds and the food that we consume. And so, I guess the other possibility is that you can find enzymes that are genes that are similar, but then one of them may not even have the function at all. And so it's a lot of kind of trying to figure out, what the key parts of the gene are that have to be conserved to predict the function.
Yeah, and I guess if they're that different, then there's also the possibility of developing really selective inhibitors, if you want to hit one but not another one.
Yeah, like it'd be great if you could keep most of your Gus activity, but only remove the activity against the drug.
So if you block that enzyme, is that going to kill the bacteria, or will they find another food source? They won't have to like flip a glucuronide off of a
yeah, that's a great question, and that's something that I'm really interested in working on more. I think in general, we don't have a great sense for why bacteria are metabolizing drugs. In the example of Gus, like the speculation is that they're using it to scavenge carbon. There's a lot of kind of classical microbiology experiments you could do to tease out how important that enzyme is, what the natural role of it is, when the drug is absent. And a lot of that still hasn't been done. So yeah, I think that's definitely a really interesting area, because it would be useful to know kind of why microbes are doing this, because then you could potentially manipulate it either to kind of drive more or less metabolism. But we don't know a lot about kind of the natural function of these enzymes.
Yeah, I mean, not even so much the metabolism, but just to kind of modify the composition of the of the microbiome so that people will be healthier in general, right?
Hmm. Right. Yeah. And even, yeah, I mean, related to the inhibition, it really makes a difference if the enzyme is critical to the growth of the microbe, because then the inhibitor becomes an antibiotic.
That's what I was thinking.
But, the kind of hope is that you can find targets that aren't that important for the fitness of the microbe. And that, that would be the ideal drug target. But the jury is still out in terms of what the kind of normal importance of these genes is. I can tell you, like, at least when I was training in grad school, the thinking was that bacteria were really stingy, and so that we typically think that bacteria don't kind of carry around genes that they don't need. I think in reality it's a lot more complicated. There's a lot of they're maybe messier than we appreciate.
So there's still a place for the classic microbiology studies. It's not all just not all genomics.
Yeah, I don't know if you saw there was just a commentary out. I think it was from Andreas Baumler at Davis, our colleague at UC, and talking about how we need more microbiologists in the microbiome field. And I totally agree with that sentiment. I say it all the time to people. A lot of people are similar to me in that they got into this because they care about genomics. And so, we have amazing genomics in the microbiome field, really elegant and impressive work that a lot of different labs are doing. But yeah, I think we've failed to kind of attract a lot of the classical microbiology people. And so that's kind of the niche that our lab has been trying to fill, is to do some of these like microbiology experiments, either in mice or in vitro.
When I first met you, I think it hadn't been too long since you published work on digoxin and the metabolism by gut bacteria. Can you talk about that? Because digoxin is a drug that's not metabolized by humans.
Mm-hmm. We got to digoxin as similar to many of the other drugs we work on. It's largely by kind of digging through the literature. So at the time I was setting up like kind of high throughput screens. Right after I started my lab, I was, working in the Harvard Center for Systems Biology as part of this Bauer Fellows program. And the goal was to try to get a better appreciation for which drugs are metabolized. But, we really lacked a lot of, and this is even like before the Redinbo paper, there weren't a lot of positive controls that we could draw from. And digoxin, as you might know, is actually in the textbook. If you read Goodman and Gilman's Bible of Pharmacology, they mention that bacteria metabolize digoxin, which I thought was so exciting.
I've forgotten that. I missed that. Okay.
The School of Pharmacy at UCSF teaches digoxin being metabolized by bacteria. So, like we've made it into the the legit pharmacology literature. But this is all before my time. It was, the drug started. A lot of these key papers came out in the 80s, kind of showing that bacteria are able to make the main metabolite, dihydrodigoxin. But they kind of hit a wall because they weren't able to really link differences in the microbes they could culture with the levels of the drug. And so, at the time they were doing this by like plating out stool samples and counting colonies. And, they were measuring the levels of these microbes that at the time were called Eubacterium lentum. We now call them Eugarbella lenta. And there was really no correlation at all with drug metabolism. And so it was an example of, we know this microbe can do this activity in vitro, but it doesn't predict anything in a patient. And so that's kind of where it stopped. And, we were fortunate enough to kind of figure out some of the microbiology. So then, I think the main goal starting out was just to find the enzyme. And the challenge was that there's no genetic tools, or weren't at the time, in Nilenta. And so we were able to confirm that digoxin is metabolized. But, how then do you find the gene? And I can remember I did an experiment that I thought was really obvious, most of the people I explained it to at the time thought it would fail. But I think it's a really useful approach that we and others have used a lot since then. We basically just gave the drug to the bacteria and then looked at what genes turned on by RNA-seq. And I think at the time I was doing a lot of RNA sequencing, so I just kind of I assumed that if you add a substrate, you're going to see more of the whatever the enzyme is. But you can imagine that would fail for a lot of different reasons. That maybe the gene is always expressed, or it's not under the control of the substrate.
Yeah, it wasn't obvious to me that would work.
Maybe I think there were, Gus maybe is an example of something that the bacteria is already expressing. I don't think they need the substrate to turn on the gene. But yeah, for digoxin it worked beautifully. It's still one of my favorite RNA-seq experiments because there's basically no gene. There's only one group of genes, this 2-gene operon that came on, and it was 10,000 fold upregulated. And so these genes are basically unexpressed, they're completely off in the absence of the drug. And then when you add the drug, they light up. And it was really specific. We didn't see any other genes affected by digoxin, to any kind of clear level. And so that really allowed us to zoom in on those 2 genes immediately. And then later on, working with a really nice, talented graduate student in Emily Belskis' lab, we were able to purify one of the proteins and show that it's actually the enzyme that acts on digoxin.
Yeah, that's fantastic. Not only did it work, but it was, you probably couldn't have hoped for a better result. That's really...
Yeah. We've still been chasing that kind of clarity of that original experiment.
Right.
For other compounds, it's been sometimes it works as advertised, but yeah, oftentimes the big issue we run into is a lot of drugs have off-target antimicrobial effects. And so if you're working in a drug like 5-fluorouracil or methotrexate, these are drugs that target conserved pathways that bacteria also need. And so if you do RNA sequencing, we see all sorts of changes that are not related at all to the metabolism of the drug.
Oh, okay, gotcha.
But the nice thing about digoxin is it really does not have antimicrobial activity that we've seen.
Okay, and that's an important one because it's a narrow therapeutic index drug.
Mm-hmm.
So if you you give a dose to somebody that doesn't have that bacteria, they might be overdosed, and you give it to somebody who does, and they might not get the benefit at all because it's all chewed up by the bacteria.
Right. Yeah, exactly. Yeah, that was definitely one of the reasons why we were excited to work on that. And also kind of what drove us into cancer. A lot of chemotherapy drugs have similar narrow therapeutic windows. And so, that's definitely a motivator, for drugs where dosing really does matter.
We organized our 2000, I guess it was 16 symposium. One of the speakers was Julia Swee, who's at the University of Washington, and she had shown some data that it was convincing to me that, and it's not just her, but she was talking about CYP3A, the host CYP3A, that it's induced by small molecule metabolites produced by gut bacteria. And to me, it was that's completely consistent with the fact that the activity of that enzyme in different people varies a lot. And to me, it makes sense that, it's induced to different extents in different people. But do you know, I mean, you're not really a drug metabolism researcher, I understand, but do you know if that is kind of accepted now? It's accepted by me, but I don't know if it's accepted in general.
Yeah, I don't know in terms of the DMPK field or industry. but yeah, I can definitely tell you in the in the literature, Julie's work and even like going way back to there's a seminal paper that Laura Hooper did when she was in the Gordon lab, where at the time they were using microarrays. But it was one of the first papers where they looked at host gene expression in the intestine in mice that were sterile versus mice colonized with specific bacteria. And she found a lot of genes relevant to drug absorption, including P glycoprotein, or the ABCB1A. Gene was actually one of the most differentially expressed genes in response to colonization. And so, yeah, I think there's a growing literature kind of showing that the expression level of these genes changes in response to colonization. But the question, I think, is how does that relate to function? Is it and that's turned out to be a little bit more complicated. We've done a little bit of work on CYP3A, but have mainly focused on P glycoprotein. And the surprising thing we found is that even though in our hands and in prior literature, colonization can induce the expression of PGP at the RNA level, we actually have found a microbe, which turns out to be the same microbe that metabolizes digoxin, that inhibit the activity of the transporter. And so we think actually the upregulation and in response to that microbe is kind of a compensatory response because the protein is getting blocked. And so I think it's really important to kind of do the next, ask whether or not the protein levels are the same, and then also like, is the protein functioning better or worse? But yeah, I think the general idea that the differences in the microbiome can control how our body handles drugs is really, that's a really wide open area that I'm really excited to see more people working.
Well, and then like the host produces bile acids, and some of those may be required, I think, by gut bacteria. Some of them may be toxic to certain gut bacteria. So it works both ways. There's a lot of communication in both directions.
Yeah, well, I think we like to think that we control the microbiome, but I guess I'm convinced it's mostly the opposite.
Agreed.
But yeah, we definitely have an immune system, and we have other kind of levers we can pull to try to maintain some sort of order.
Oh man, we could talk for hours about the effect of the gut microbiome on the immune system, and the gut-brain axis, but we'll try to stick to DMPK.
The most important area of microbiome.
There you go. So, one class of drugs that is just really huge right now are the GLP-1 drugs, semaglutide, and drugs like that. Do those have do we know if there's any interaction between those and the gut microbiome, if they affect the gut microbiome or vice versa?
Yeah, it's definitely an area I'm really interested in. We didn't really get into this, but the thing I actually worked on when I was in grad school was diet and obesity, with and how it relates to the microbiome. And then when I kind of started my own lab, I switched into mainly thinking about pharmacology. But we've continued to think about diet and metabolic disease throughout the years. And the frustrating thing has been there's never been a drug for a drug that really worked well for obesity.
Right.
And so I'm really excited now that these drugs seem to be working, and that allows us to kind of think about pharmacology and with respect to diet and obesity. There's not a lot out there, and surprisingly to me, given that there's been really massive human studies of these drugs, and so nowadays I would expect that most of these clinical trials would have some sampling to look at the microbiome, but that really hasn't been published. There's been some smaller studies in mice that have found something I think somewhat surprising, which is that these drugs really do seem to change the microbiome. And that's not really well understood why that would be the case. And then in addition to that, there is a decent literature that kind of predates these drugs, including really nice work from Frederick Fackhead, another former Gordon Lab member, um, looking at how the microbiome controls GLP release. And that's I think a really interesting and related area, kind of how the kind of baseline levels of GLP-1 and other incretins are controlled by microbes, and that might kind of set the baseline for how these drugs are acting on people.
Yeah, or maybe some people don't need them; they just need a healthier gut microbiome.
Could you get the effect without the drug? Yeah, I think it'll be interesting to see kind of how far the microbes can push it, as opposed to these, agonists that are incredibly potent. But,
Okay, yeah, I'll have to keep my eye on that. That's interesting that it hasn't really been published yet, but I guess somebody must be working on that.
Yeah, maybe. Yeah, if you know anyone at Novo or, or the other companies, I'm happy to talk to them.
Yeah.
I guess more generally, I'm always surprised when like companies invest so much. At this point, microbiome sequencing is pretty cheap, and so it's weird to me that you would go to the trouble of doing a large scale phase 3 trial without throwing in some microbiome sequencing, even on a subset of the cohort.
Yeah, especially with these drugs, I know that because there are side effects, GI side effects for sure.
Yeah, like even at a minimum, you might want to look at the microbiome of the people that have GI symptoms. But maybe it's hard to know. Maybe it is happening, and we just haven't heard about it. But I don't have any kind of insider knowledge.
Right, or the company got an answer they didn't really want.
Right.
Wasn't the answer they were hoping for.
Like, yeah, that really changed the microbiome. Yeah, I mean, related to that, I think that the hard part, I think, for industry and as well as for academics is like, it's hard to say what the consequence of a change in the microbiome is. And so, it's very easy to do the study to measure changes in the microbiome in response to a drug. But then, the question you might get from, the FDA or from other folks at the company is like, is this change good or bad? And that's something that's actually quite difficult to answer. And so, I think maybe part of the hesitancy is, they don't really know what they would do with the data if it were collected.
Yeah, because it's not so much about bacteria that are good or bad. It's more about the balance of all of them.
Right.
Yeah.
Yeah, and I think really more specifically what you care or what I would care about is like, are those changes, do they matter for the direct mechanism of action of the drug, or do they matter for the side effects of the drug?
Yeah.
And that's something that you really have to do more experiments to figure out. We don't really have a way to just look at the list of microbes and tell you the answer.
So I wanted to ask you something that maybe some of our listeners are wondering about, but I'm definitely wondering about. Is it really feasible to directly study bacterial drug metabolism in vitro? You mentioned, we talked about the fact that the bacteria we'd be interested in are anaerobic, and you said there are chambers where you can work with them, but we'll take microsomes or liver microsomes out of the freezer and do a drug metabolism study, metabolic stability, and measure metabolites. Is it that simple with gut bacteria too?
Yeah, I mean, I think the good news is that, with some exceptions, most of the microbes can be cultured. So then it's just mainly logistics and like, do you have the right equipment? Do you have personnel that are comfortable growing anaerobes? But it's not really rocket science, and that it's mainly using special chambers that keep them away from oxygen. I think maybe the question, related question that you're alluding to is whether or not the rate or extent of metabolism in vitro, can be extrapolated to an in vivo PK experiment. That is a wide open question, and it's something we really don't have a great handle on. And I definitely think what we've learned over the last few, 10 to 15 years has kind of complicated how you would imagine going about that, because, in the simplest model is that the only thing happening is this direct metabolism. But as we've been discussing, the same microbe that metabolizes digoxin can also control the transporter P glycoprotein that influences absorption of digoxin. And while the transporter effect seems to be conserved among all of Elenta species, only certain strains are able to metabolize the drug. And so, you kind of have to think about the specific strain of microbe that's in the community, and also these like more indirect ways that it could be influencing the drug. And so I think that suggests that it's not going to be a one-to-one where, you won't be able to say, this microbe was active in vitro. I would expect exactly this amount of change in PK.
Yeah, not even, I wouldn't be thinking trying to scale it quantitatively, but more just like, I have this new drug candidate. Could it be metabolized by gut bacteria? And, what metabolites might I look for? And I mean, can you?
Yeah, I think to that, that question is much more answerable. So the main challenge there, I think, is just the diversity of microbes. So like, as opposed to the liver where there's a finite number of enzymes, there's always another microbe to test. And so, yeah, I think we in general try to avoid saying that the microbiome can't do something, because it might just be we haven't looked at the right microbe. But you can definitely, you can within reason, screen 100 microbes or, a set of complex microbial communities, and then see whether or not you find anything or not. And if you find metabolites, then you know that those can be produced by microbes.
So I feel like I'm looking at this so naively, but like, would it be useful to start with fecal samples, fecal homogenates, or are those all dead bacteria? Are there enough live ones that you would get an idea of what might be happening in the gut?
Yeah, no, it's not naive. Like, yeah, this is we call it, well, there's a lot of different words for this, but we like to call it ex vivo incubations, which, sounds cleaner. People do other stuff like you can, so in the way that we do ex vivo incubations, you're just diluting the cells into media and then incubating. And so that I think is a useful way to ask whether or not this complex community that you just pulled out of a human is doing something to a drug. The challenge is that as you culture, I think we talked about way at the beginning, is that, like, the culturing process is going to change the community. And so we currently don't have a way to kind of fix the community in place, maintain the same structure that it was in the patient. And so that leads to all sorts of confusing results where you have expansions of different microbes that you don't want in your sample. And so one solution for that, I think they call it fecalaise, but you can just take a stool sample and lyse all the cells, so that what's left is just a big collection of enzymes, and then test that for activity. And so and then there's different medias that people use, and there's various options kind of within those two major categories.
And like how rigorous do you have to be about excluding oxygen? Like, from the initial sample and all the way through processing and shipping and storage.
Yeah,
exclude oxygen?
it's a good question. We definitely think it's important. And yeah, this maybe won't make you want to do this approach. I mean, one of the nice things about these like DNA-based profiling methods is that they tend to be less sensitive to that because the DNA is really stable.
Yeah, right.
And so, if you're doing sequencing, you don't have to worry about that as much. We, a while ago now, had a paper where we were developing methods of kind of measuring live and dead bacteria within a sample. And for those metrics, it was really important that the sample got straight into the anaerobic conditions. So even, within hours, you can start to see a lot of the cells dying. And that makes sense given that the most abundant members of the human gut microbiota are also very oxygen sensitive. So, I think that's probably one of the reasons why, like, even if you're culturing under anaerobic conditions, on petri dishes, we often find, the usual suspects like E. coli and Enterococcus and, all of the microbes that can handle oxygen.
Yeah. Okay. Yeah. You unintentionally select for those.
Right. Yeah. Just like it's really hard if you're thinking about a clinical study because then you're typically sampling patients that are not even near the lab. And so, as opposed to collecting a local sample. Or in mice, we have the advantage of being able to actually, you can even bring the organ into the anaerobic conditions. Yeah.
Okay. Peter, this has been a fascinating and insightful discussion, and I thank you so much for your time. Um, really, really appreciate it.
Sure. Yeah, no problem. It was really fun.
So thank you all for listening to this episode of Pharmaron's DMPK Insights podcast series. We would like to remind you that our DMPK webinar series is also available on demand, covering a variety of key questions related to DMPK science and drug discovery and development. And stay in touch for more episodes in the Pharmaron DMPK Insights podcast series. Thank you and goodbye for now.